Hello, I'm Linda. I'm a postdoc at the University of Toronto, and today my talk is entitled Reconstructing Tumor Evolutionary Histories and Clone Trees and Polynomial-Time with SubMARine. Tumors evolve by accumulating mutations over time, leading to a heterogeneous set of genetically distinct cancer cells. Knowing about the evolutionary history of a tumor can help us understand how it developed and how it might respond to treatment. Given the sequencing data of a tumor sample taken at a specific time point, different methods exist to infer the frequencies of so-called subclones, from which clone trees can be built. A clone tree is a directed rooted tree whose nodes correspond to subclones and whose edges link parental subclones to their direct descendants. A subclone is a group of cells that descended from the same founder cell and represents to the subtree in the phylogeny of cancerous subpopulations. Each subclone is associated with a set of mutations and a subclonal frequency phi. This frequency corresponds to the frequency of subclones in the cancer sample that was taken. Given a subclonal frequency matrix phi, there exist two key challenges. The first challenge is scaling algorithms to a large number of subclones, and the second challenge is efficiently representing uncertainty in a reconstruction. In order to address these two challenges, we've developed SubMARine, a polynomial-time algorithm that builds partial clone trees. The notion of a partial clone tree is a main contribution of our work. A partial clone tree defines a subset of the ancestral relationships between subclones. It defines when a subclone is an ancestor or not an ancestor of another subclone. And it may also contain undefined relationships, when a subclone is a possible ancestor of another subclone. We represent these three different kinds of relationships in an ancestry matrix. A visually more intuitive representation of this partial clone tree is this possible parent DAG, where we show when a subclone is a parent, is not a parent, or is a possible parent of another subclone. As you can see, a partial clone tree is not a tree itself, but implicitly represents a set of clone trees. This is the set of trees that is consistent with the defined relationships in the partial clone tree and that chooses their parents from the possible parents. So for our example, these two clone trees are the completing clone trees, where one subclone 4 chooses subclone 2 and one subclone 1 as parent. Given the subclonal frequency matrix phi, for which a set of valid clone trees exists, the maximally constrained ancestral reconstruction, or short MAR, is a special partial clone tree that represents efficiently the uncertainty in the solution set. It defines the maximal set of all of the ancestral relationships shared by the solution set of the clone trees, and leaves undefined all relationships that vary between those clone trees. Given the solution set, it's trivial to build the MAR. However, the solution set is usually not given. This is why we developed SubMARine, our polynomial-time algorithm that approximates the MAR by constructing the subMAR, which is a partial clone tree that has three important properties. First, the subMAR is unique, which means that given the subclonal frequency matrix phi, there's only one single subMAR for this dataset. The second property is that the defined ancestral relationships in the subMAR are a subset of those in the MAR. That means whenever SubMARine defines a relationship, we know that this relationship also has to be defined in the MAR. On the other hand, if SubMARine leaves the relationship undefined, we cannot be sure whether it does not have to be defined in the MAR. From these two properties, the third property follows immediately, which is that all valid clone trees for the input data phi are completions of the subMAR. So, as I said, SubMARine takes the subclonal frequency phi as input, and it then builds the partial clone tree represented in the ancestry matrix Z, where initially all relationships are undefined. It then defines relationships by propagating a set of validity constraints for partial clone trees. Then, in the end, it outputs the ancestry matrix Z and the possible parent matrix tau. If you want to hear more about these rules and how the propagation works, you can look at the poster presentation that I presented at ISMB. Next to the basic version of SubMARine, we also implemented an extended version that takes copy number operations as input. We applied SubMARine on a large simulated dataset with 2,400 clone tree reconstruction problems with up to 50 clone trees, and on the TRACER-X lung cancer study with 88 patients and up to 15 subclones. The subclonal frequencies in the simulation were noise-free, while they contained noise in the TRACER-X study. SubMARine was super fast. On the simulated data, it needed less than 70 seconds to construct each subMAR, and it needed less than 40 seconds on the TRACER-X data. Note that for 3 patients in this dataset with more than 10 subclones, the authors were not able to run sit-up. That's the method that they use to build clone trees. So they had to build the clone trees manually. As we could see on the simulated data, the more constrained the input data is, the less undefined relationships we have in the partial clone tree. You can see this in these 2 plots, where on the y-axis we have the proportion of subclones that have multiple possible parents, so that are directly affected from undefined relationships, and on the x-axis we have the number of samples. And as you can see, this proportion of subclones decreases with the number of samples that poses more constraints on the ancestry matrix. And also on this right side, where the input data had copy number aberrations, there's less uncertainty in the clone tree. On the simulated data, we could also see that the more constrained the input data was, the more the subMAR actually resembled the MAR. The subMAR actually matched the MAR in 93.9% of the time. And for a MAR where there was only a single valid clone tree solution, the subMAR equaled the MAR in 99.9% of the time. In the TRACER-X data, the subMAR always equaled the MAR. And for the TRACER-X data, we also compared the clone trees completing the subMAR with the clone trees reported in the TRACER-X study, and the subMAR represents all those trees in 73.9%, and it represents a subset of those trees in 21.6% of the time. So you can see that the results largely resemble those results reported in the TRACER-X study. So in this work, we introduced the notion of a partial clone tree. Our clone tree reconstruction method, SubMARine, scales to a large number of subclones and efficiently represents uncertainty in the subMAR. We've seen that the more constrained the input problem is, the more the subMAR resembles the MAR. You can find SubMARine on GitHub, and you can have a look at our preprint on bioRxiv. And now last but not least, I'd like to thank Jeff, Gunnar, Jens, and Quade, who worked with me on this project. And I also like to thank you for listening. I'm happy to answer questions, and you can also come to the poster session if you want to learn more about SubMARine. Thanks.